Hello, my name is Brittany Richardson Michael, and today we're going to talk about cell cycle regulation in the cell. So, before we begin, I would like to discuss the objectives of this presentation today. Understand the importance of cell cycle regulation in maintaining cellular homeostasis and preventing uncontrolled cell proliferation. Identify key regulatory checkpoints in the cell cycle, including G1S, G2M, and metaphase-anaphase transitions. Explain the role of cyclin-dependent kinesis, or CDKs, and their associated cyclins in driving progression through the cell cycle. And finally, discuss the functions of tumor suppressor genes such as P53 and RB in monitoring DNA integrity and halting cell cycle progression in response to damage. Okay, so let's dive right in. What is the purpose of the cell cycle? The purpose of the cell cycle is to ensure that cells grow, replicate their DNA, and divide into two daughter cells. This process is essential for growth, development, tissue repair, and reproduction in organisms. According to Alberts, the cell cycle is a series of events that take place in a cell leading to its division and duplication. The cell cycle consists of several phases, including interphase, which is G1S and G2, where the cell grows and replicates its DNA, and mitosis, where the cell divides into two identical daughter cells. Overall, the cell cycle plays a crucial role in maintaining proper cell function and ensuring the accurate transmission of genetic information from one generation of cells to the next. What is cell cycle regulation? As you see in the picture that I have up, you have interphase, prophase, metaphase, anaphase, telophase, and cytokinesis. Cell cycle regulation refers to the control of the progression of a cell through various stages of the cell cycle, including interphase, mitosis, and cytokinesis. This regulation is crucial for maintaining proper growth. Maintaining proper growth, development, and function of cells in an organism. Dysregulation of the cell cycle can lead to various diseases, including cancer. One key aspect of cell cycle regulation is the activity of cyclin-dependent kinesis, or CDKs for short, and their regulatory subunits called cyclins. The activation and inhibition of CDK cyclin complexes at a specific point in the cell cycle drive progression from one phase to another. Additionally, other regulatory molecules such as checkpoints and tumor suppressors help ensure that DNA replication and cell division occur accurately. According to Lodish, the eukaryotic cell cycle is controlled by a series of protein kinesis called cyclin-dependent kinesis. These CDKs are activated by binding to specific proteins known as cyclins at different stages of the cell cycle. What are the phases of the cell cycle? So as you see on the image, you have interphase and mitosis. So in interphase, the cell grows and copies its DNA. You have G1S and G2, which is GAP1 synthesis and GAP2. In G1, the cell is the cell growth. S or synthesis is DNA synthesis. G2 is more growth and preparation for mitosis. Now, in mitosis, the cell divides its DNA and cytoplasm and forming two new cells, or the two daughter cells. In mitosis, you have prophase, you have metaphase, anaphase, and telophase. And as you see, you have a G0 at the bottom. And this is just the resting state where the cell performs its functions, but it is not preparing to divide. So the cell cycle is a highly regulated process that consists of distinct phases, each with specific functions and checkpoints to ensure accurate replication and division of the cell. The cell cycle can be divided into four main phases, which are the phases that we discussed on the image, and that's G1, S, G2, and mitosis. During the G1 phase, the cell grows in size and synthesizes proteins necessary for DNA replication. The S phase is when DNA synthesis occurs, resulting in the duplication of the genetic material. In the G2 phase, the cell continues to grow and prepare for mitosis by synthesizing additional organelles and proteins needed for cell division. And finally, in mitosis, which is the M phase, it includes mitosis and cytokinesis, which is the duplicated chromosomes are separated and distributed to two daughter cells. Lotus stated that eukaryotic cell cycles is contributed by a complex network of regulatory proteins including cyclins, CDKs or cyclin-dependent kinesis, and checkpoint proteins. These regulators ensure that each phase of the cell cycle is completed accurately before progressing to the next phase. Disruption of these checkpoints can lead to abnormal cell division and potentially contribute to diseases such as cancer. What is the importance of cell cycle regulation? Proper regulation of the cell cycle is crucial for maintaining genomic integrity and preventing diseases such as cancer. Dysregulation in the cell cycle can lead to uncontrolled cell proliferation and tumor formation. Cell cycle regulation is crucial for maintaining the proper growth, development, and functioning of cells in an organism. It ensures that cells divide at the right time, in the correct order, and with the appropriate frequency to maintain tissue homeostasis and prevent diseases. The cell cycle is controlled by a complex network of proteins that regulate the progression of a cell through different phases of the cell cycle. Dysregulation of the process can lead to uncontrolled cell division, genetic instability, and ultimately formation of tumors. Proper regulation of the cell cycle also plays a key role in the embryonic development, tissue repair, and immune response. So, for example, during embryogenesis, precise control over the timing and coordination of the cell division is necessary for the formation of organs and tissue. In adult organisms, regulated cell proliferation is essential for wound healing and immune response to pathogens. Understanding and controlling cell cycle regulations are critical for maintaining cellular health and preventing diseases. By studying the mechanism involved in regulating the cell cycle, research can develop targeted therapies to treat various disorders associated with the abnormal cell division. What are key players in cell cycle regulation? And as you see, I have bulleted down here cyclins, cyclin-dependent kinesis or CDKs, and their inhibitors, which are P21 and P27. So, key players in cell cycle regulation include cyclins, cyclin-dependent kinesis or CDKs, and their inhibitors such as P21 and P27. These components play crucial roles in driving the progression of cells through the different phases of the cell cycle, including anaphase, which is G1, S, and G2, and mitosis. Cyclins are regulatory proteins that bind to CDKs to activate them at specific points in the cell cycle. CDKs are a family of protein kineses that phosphorylate target proteins to regulate various cellular processes, including progression through the cell cycle. The activity of cyclin CDK complexes is tightly controlled by both positive and negative regulators, such as a cyclin-dependent kinesis inhibitor or CKIs, like P21 and P27. These CKIs act as brakes on the cell cycle by inhibiting the activity of cyclin CDK complexes, thereby regulating cell proliferation. According to Sher CJ, Cell Cycle Control Center, cells possess two families of CDK inhibitors that function at the specific point in the cell cycle and prevent unscheduled DNA replications or centrosome duplication. So overall, the coordination of cyclin CDKs and their inhibitors is essential for the proper cell cycle progression and maintenance of genomic stability. Now let's talk about the control system. What is the cell cycle control system? So as you see in the image, you have the control system. You see you have G1, G2S, mitosis, and you see on the outskirts you have the orange things popping up, and you see those are checkpoints. The cell cycle control system is a highly regulated process that ensures the accurate duplication and division of cells. It consists of a series of checkpoints that monitor key events throughout the cell cycle, allowing for corrections and adjustments to be made if necessary. These checkpoints are crucial in maintaining genomic integrity and preventing the proliferation of damaged or abnormal cells. The cell cycle control system includes a complex network of regulatory proteins and signaling pathways that coordinate the progression of cells through each phase of the cell cycle. The main checkpoints in the cell cycle control system are located at the G1/S phase or transition, S phase, and G2/M transition. At these checkpoints, various proteins such as cyclins, CDKs, cyclin-dependent kinesis, and tumor suppressor genes like P53 play critical roles in regulating cell cycle progression. So as you see, it has to be a checkpoint to make sure everything is doing what it's supposed to do. Everything is happening in each phase the way it's supposed to be. And if not, if it's caught early on, adjustments can be made. This regulation of the cell cycle control system can lead to uncontrolled cell growth and contribute to the development of diseases. Therefore, understanding how the system operates and identifying potential targets for intervention is essential for developing effective therapeutic strategies. There is ongoing research to uncover new insight into the mechanism governing the cell cycle control system. And that's offering promising opportunities for the novel treatments and various pathological conditions. What is the role of checkpoints in cell cycle regulation? So previously we discussed the checkpoints. Where are the checkpoints? What are the checkpoints? Now we're going to discuss the role of those said checkpoints. So as you see on the image, you have the phases, and then you see it has mitosis. You see you have cell growth checkpoint, you have DNA synthesis checkpoint, and you have mitosis checkpoint. As you see the green arrow that says G0, that is where it's resting. Nothing is getting ready to split or anything. It's resting. So checkpoints play a crucial role in cell cycle regulation by ensuring that each phase of the cell cycle is completed accurately before progressing to the next phase. Um, these checkpoints serve as a control mechanism that monitor key events such as DNA replication, chromosome segregation, and cell size, allowing cells to maintain genomic integrity and prevent the accumulation of mutations. Checkpoints controls Checkpoint controls ensure that each event in the cell division cycle is completed accurately before progression occurs to the next event. There are several major checkpoints in the cell cycle, including the G1S checkpoint, the G2M checkpoint, and metaphase to anaphase checkpoint, the metaphase to anaphase transition checkpoint. So the G1S checkpoint monitors DNA integrity before entry into the S phase, while the G2M checkpoint ensures proper DNA replication and repair prior to entering into mitosis. So as you see, it's steps that you gotta take. Additionally, the metaphase to anaphase transition checkpoint verifies correct alignment of chromosomes on the metaphase plate before anaphase onset. So it's like checkpoints inside of checkpoints. If errors or abnormalities are detected at any of the checkpoints, the cell cycle can be arrested temporarily to allow the necessary repairs or corrections. So it halts for a little while to be fixed, fix. Failure to properly regulate these checkpoints can lead to uncontrolled cell proliferation and potentially result in cancer development. Therefore, checkpoints play a critical role in maintaining cellular homeostasis and preventing cell division. So on the picture, as you see, the checkpoints is broken down with cell growth checkpoint, which is the first checkpoint, and it occurs towards the end of the growth phase 1 and it checks whether the cell is big enough and has made the proper proteins for the synthesis phase. If not, the cell goes through a resting period until it's ready to divide. So, yes. In the DNA synthesis checkpoint, this occurs during the synthesis phase or the S phase, and it checks whether DNA has been replicated correctly. If so, the cell continues on to mitosis. And 3, the mitosis checkpoint occurs during the mitosis phase, and it checks whether mitosis is completed. If so, the cell divides. So what are cyclins? I know y'all done heard me say cyclins, CDKs, say y'all not probably not. What is a cyclin? So cyclins are a family of proteins that play a role in regulating the cell cycle, specifically by binding to and activating cell cyclin-dependent kinesis, or the CDKs y'all constantly heard me say. This interaction between cyclins and CDKs is essential for controlling the progression of cells through different phases of the cell cycle, including G1S and G2 and mitosis phase. The levels of cyclin fluctuate through the cell cycle, with their expression peaking at specific points to ensure proper coordination and cell division. According to Lodish, cyclins are so named because they accumulate during anaphase and are abruptly destroyed as cells enter mitosis. Different types of cyclins are expressed at different stages of the cell cycle, allowing the precise regulation of CDK activity. For example, cyclin D is involved in regulating progression from G1 to S phase, while cyclin B is essential for entry into mitosis. Cyclins are key regulators of the cell cycle that works in conjunction with CDKs to ensure accurate and timely progression through each phase. Their dynamic expression patterns highlights their importance in maintaining cellular homeostasis and preventing uncontrolled proliferation. What are CDKs or cyclin-dependent kineses? CDKs or cyclin-dependent kineses are a family of serine protein kineses that play a crucial role in regulating the cell cycle. CDKs work in conjunction with their regulatory subunits, cyclins, to phosphorylate target proteins involved in various stages of the cell cycle, including DNA replication, chromosome condensation, and cell division. CDK activity is tightly regulated through association with specific cyclins and by phosphorylation at multiple sites. The, act, the activation of CDK cyclin complex triggers the progression of the cell cycle by promoting the transition between different phases, such as G1S and G2M checkpoints. Additionally, CDK inhibitors, CKIs, act as negative regulators of CDK activity to prevent uncontrolled cell proliferation and maintain genomic stability. Dysregulation of CDKs has been implicated in various diseases, including cancer, where cell cycle control leads to unchecked cell growth and tumor formation. CDKs are inter- CDKs are integral components of the cell cycle machinery that govern the orderly progression of cells through different stages. Their precise regulation ensures proper cellular division and function, highlighting their significance in maintaining tissue homeostasis and preventing pathological conditions. Now let's talk about the regulation of gene expression. So transcription factors and signaling pathways regulate gene expression during the cell cycle. These mechanisms control the activation or repression of genes involved in cell cycle progression. Regulation of gene expression is the process by which a cell controls the synthesis of protein based on the information encoded in its DNA. The regulation ensures that genes are turned on or off at the right times and in the right amounts to maintain cellular function and respond to environmental cues. There are several levels of regulation involved, including transcriptional control, post-transcriptional control, translational control, and post-translational control. Transcriptional control is a key step in regulating gene expression where specific transcriptions factors bind to regulatory regions of DNA to either activate or repress gene transcription. Transcriptional regulation allows cells to express different sets of genes under different conditions. Post-transcriptional control involves processes such as RNA processing, splicing, and stability that can influence mRNA levels. Translational control occurs when regulatory molecules affect the initiation or elongation of stages of protein synthesis. Lastly, post-translational modifications can alter protein function after translation. The precise regulation of gene expression is essential for maintaining cellular homeostasis and responding to changes in the internal or external environment. Understanding these regulatory mechanisms provides valuable insight into how cells function and how they adapt to various stimuli. External factors influencing cell cycle regulation. The cell cycle is tightly regulated by both internal and external factors. to ensure proper growth and division of cells. External factors play a crucial role in influencing cell cycle regulation. These external factors can include signals from the neighboring cells, environmental conditions, and extracellular cues. One key external factor that influences cell cycle regulation is the availability of nutrients. The process or absence of essential nutrients such as amino acids, glucose, and growth factors can greatly impact the progression of the cell cycle. Cells require adequate nutrients to support their metabolic processes and DNA replication during the cell cycle. A lack of nutrients can lead to cell cycle arrest as cells prioritize survival over proliferation. In addition to nutrient availability, external signals from neighboring cells also play a significant role in regulating the cell division by activating signal pathways that promote entry into the cell cycle. Conversely, conversely, signals from neighboring cells can induce cell cycle arrest to prevent uncontrolled proliferation. External factors such as nutrients available and signaling from neighboring cells are critical in influencing cell cycle regulation. Diseases associated with dysregulated cell cycle. Dysregulated cell cycle control can lead to the development of various diseases, including cancer. Cancer is characterized by uncontrolled cell growth and division, which results from abnormalities in the regulation of the cell cycle. Deregulation of the cell cycle machinery is a common event in human cancer. When key regulatory checkpoints in the cell cycle are disrupted, cells may continue to divide uncontrollably, leading to tumor formation. In addition to cancer, dysregulated cell cycle control has also been associated with other diseases such as neurodegenerative disorders and autoimmune diseases. For example, cell cycle progression in neurons has been linked to neurodegenerative conditions like Alzheimer's or Parkinson's disease. Furthermore, defects in immune cell proliferation due to the dysregulated cell cycle control can contribute to the development of autoimmune disease such as lupus and rheumatoid arthritis. Overall, understanding the mechanism underlying dysregulated cell cycle control is crucial for the pathogenesis of various diseases. In conclusion, the cell cycle regulation in cells is a highly intricate process that involves multiple checkpoints and regulatory mechanisms to ensure proper cell division and growth. The key players in the process include the CDKs, cyclin, and various checkpoints proteins that monitor DNA integrity and cellular conditions. Dysregulation of the cell cycle can lead to uncontrolled cell proliferation, which is a hallmark of cancer development. The cell cycle has been the focus of intense scrutiny for many years because its misregulation leads to diseases such as cancer. Understanding the mechanisms underlying cell cycle regulation is crucial for developing targeted therapies for cancer treatment and other related diseases. The study of cell cycle regulation continues to be a dynamic field with ongoing research aimed at unraveling the complexities of this essential biological process. Thank you guys for your time. I truly hope you guys enjoyed the presentation.